Let's now take a look at the process of gastrulation in amphibians. The major types of movements that occur during amphibian gastrulation are summarized in this image. These include epibole, which is the thinning and the spreading of the animal cap cells over the vegetal hemisphere, and that's powered by proliferation of the cells and a process known as radial intercalation, which we'll discuss in a moment. There's also vegetal rotation, where the vegetal cells asymmetrically press up against the inner blastocele roof on the dorsal side. Some cells, which are known as the bottle cells because of their shape, begin to exhibit an apical constriction, which promotes invagination. Gastrulation in amphibians is also characterized by involution and cell migration, which allow the leading edge of invaginating cell to crawl onto the blastocele roof. Finally, the process of convergent extension, which involves the intercalation of cells in the midline, drives the elongation of the anterior-posterior axis. The movement of cells during gastrulation begins at the dorsal side of the embryo in a region that is known as the marginal zone, which is located slightly below the equator of the egg at the point where the animal and vegetal poles meet. This is the region where the gray crescent becomes exposed as a result of the cortical rotation that happens after sperm entry. So this is the portion of the egg that's located at the opposite side of sperm entry. At this site, some of the cells elongate and begin to invaginate, forming a blastopore that initially looks like a slit. The blastopore is the point in a gastrulating embryo at which cells are moving inward to form the primitive gut or archenteron. In deuterostomes like amphibians and mammals, the blastopore becomes the anus. In protostomes, the mouth forms at or near the blastopore opening. As depicted in these diagrams, the elongated cells, which are known as the bottle cells, extend their body toward the inside of the embryo while still remaining in contact with the outer surface of the embryo. These cells set into motion the formation of the archenteron or primitive gut. The next phase of gastrulation involves the involution of the marginal zone cells, while the animal pole cells undergo epibole and convergence at the blastopore. Once the marginal zone cells come into contact with the wall of the blastocele, these cells begin to involute into the embryo. They travel along the roof of the blastocele, coming in contact with the cells of the animal hemisphere. The order in which the cells enter into the embryo is determined by a series of early events that are collectively known as the vegetal rotation. This vegetal rotation takes place about 2 hours before the formation of the bottle cells. It involves the movement of cells of the dorsal floor of the blastocele toward the animal cap. As a result of these movements, the cells that are going to give rise to the head endoderm, which is known as the pharyngeal endoderm, are placed adjacent to the blastocele and above the mesoderm as it begins to involute. Eventually, as you can see in this image, the cells of the head endoderm lead the way, migrating along the inner surface of the roof of the blastocele as they travel to what's going to become the future anterior of the embryo. At the same time, the animal pole cells undergo epibole, producing a stream of cells that converge at and become the dorsal blastopore lip. Cells located in the region of the marginal zone get pulled into the embryo and form the lining of the archenteron or primitive gut. It's worth noting that if these cells are removed, the archenteron does not form properly. As I mentioned previously, the cells that lead the way into the embryo migrating toward the anterior and under the surface ectoderm are those that will give rise to the head endoderm, which is also known as the pharyngeal endoderm. These cells express the transcription factor H hex, which is required for the formation of the head and the heart. Once these cells enter into the embryo, the identity of the dorsal blastopore lip changes as it is now composed of cells that will give rise to the head mesoderm, and these cells transcribe the glucocorticoid gene, which is a transcription factor that indirectly activates the genes required for head formation. The next cells to involute are those that will give rise to the notochord, which is a transient rod of mesoderm that induces the development of the nervous system. These cells express the transcription factor brachyury, which is required for the formation of the notochord. So as you can see, the cells that make up the dorsal blastopore lip are constantly changing as the original cells migrate into the embryo and are replaced by cells migrating downward, inward, and then upward. This temporal change in the cells that make up this structure is important for a series of inductive interactions that we'll encounter later. As involution continues during gastrulation, the lip expands laterally and ventrally. So the initial slit-like dorsal blastopore lip widens and develops ventral and lateral lips through which additional endodermal and mesodermal precursor cells pass. So the blastopore goes from a slit to a ring that surrounds large endodermal cells of the vegetal hemisphere. That patch of endoderm that remains exposed is known as the yolk plug. Eventually, the yolk plug is internalized and is the site that will give rise to the anus of the organism. The process of convergent extension plays a key role in gastrulation and in other morphogenetic processes as well. It's a mechanism for elongating a sheet of cells in one direction while narrowing its width, and it occurs by the rearrangement of cells within a sheet. It can happen by two different mechanisms, and one of these involves the intercalation or shuffling of cells between each other along the mediolateral axis. The molecular mechanisms that control convergent extension involve the activation of the Wnt planar cell polarity pathway, which leads to changes in the cell's actin cytoskeleton. Additionally, the expression of different cadherin types is necessary for cells that will give rise to different mesodermal structures to be able to sort out from each other. A mechanically similar process called radial intercalation causes the thinning of a multicell layer into a thinner sheet of cells. Radial intercalation occurs in the multi-layer ectoderm of the animal cap, and in this process, cells intercalate in a direction that's perpendicular to the surface. So that leads to an increase in the surface area of the cell sheet, as well as the thinning of the sheet, and this is in part responsible for epiboly. In addition to radial intercalation, the process of epiboly also requires the expression of fibronectin. The assembly of fibronectin in the ECM of the cells along the roof of the blastocele is needed for cells of the animal cap to surround the embryo and to enclose it. Additionally, the involuting mesodermal cells migrate by traveling on a network of fibronectin that's secreted by the presumptive ectodermal cells along the roof of the blastocele. This network of fibronectin is labeled in yellow in panel B. If embryos are incubated with a peptide that binds to fibronectin binding sites in the cells, then the cells are unable to migrate and the presumptive mesoderm remains outside of the embryo, as shown here in figure D. So the driving forces for ectodermal epiboly and for the convergent extension of the mesoderm are the intercalation events in which several tissue layers merge. And fibronectin plays a critical role in enabling the mesodermal cells to migrate into the embryo. Take a look at the following videos which depict the process of gastrulation and neurulation, which is the formation of the neural tube.